When I sit down, and now I will do it. There.
There's like 42 people.
This old lady got to sit down.
Okay, down a little bit.
Hey, everybody.
Okay, there you go.
There you are.
You're
Looking fine.
Watch
Okay.
Everything looks really
we
good.
are recording. We are now recording, everybody. Watch your language, please.
Watch our language. Okay.
Here we go. Thank you.
You think the recording is going to make any difference in our language? You're sadly mistaken.
You Archie.
86 ATJ Marine Mobile.
86 Old Buckingham.
This is so cool.
Well, this is the fun part.
This is.
Hi, everybody. Unfortunately, I just did my power thing and I muted everybody because it's time just about for us to start our program. And we already have 33 people online, and I'm sure many more coming on stream very shortly. So I would, on behalf of the board of directors, welcome all of you to our second monthly virtual PEP luncheon. And I hope you enjoyed the lunch. The salad bar, I know you enjoyed. I saw many of you up there, and I thought the Caesar salad was excellent today. Ruby has been a little bit overworked, and but she's there, and she's will be serving cookies and the ice cream sundaes very shortly. Just to review the agenda for everybody, we're going to start at about 11:30, and that's coming right up quick. And Dr. Rossiter will speak, and I'll formally introduce him in a minute. After he speaks, he'll speak from 11:30 to around 12:15. And during his speech, he has the capabilities of sharing some documents on screen. So don't freak out if he pops up and has a PowerPoint slide on screen. That's what he's planning on doing. And he'll speak from 11:30 to 12:15.
Hey, it's me.
And
The Steinway is going out of business.
12:15,
I'll talk to you later.
we will open it up to Q&A. And we'll go from 12:15 to 12:30 for our Q&A. It's so great to have Dr. Rossiter join us. I had the privilege of working with him at the Pulmonary Education and Research Foundation, PERF. He's a great guy, and I wanna tell you a little bit about him because he's no a stranger to our field of cardiopulmonary obstructive disease. He is an investigator at the Lundquist Institute. He is an associate professor of respiratory and clinical care physiology and medicine at the David Geffen School of Medicine at UCLA. He is also director of research training, Division of Respiratory Critical Care Physiology and Medicine at Harbor UCLA Medical Center. He is director of the UCLA CTSI High Resolution Tissue Respirometry Core at LA Biomed. And I gotta tell you, nobody knows more about exercise and the impact on people with COPD than Dr. Rossiter. So it really is my privilege and honor to introduce our guest speaker, Dr. Harry Rossiter. It's all yours, Harry. Give him a round of applause, please.
Thanks so much, Kurt, and everybody. It's just amazing to see you all. I love coming to the Sizzler, and unfortunately that disappeared, and now we have COVID. So this is how we have to do it. But you know, we'll look forward to getting back to something more normal soon. I'm going to try and share my screen here.
Ooh.
How's that? Can you see, everyone see my title slide?
Yes, we can, Harry.
Good. Then it's working. That's excellent. Let me see. I've got to move you guys out of the way so I can see what I'm doing. There we go. Thanks to Kurt and Jackie and others for inviting me to talk with you today. I'm going to talk about our favorite topic, COVID-19, and its impact that it's had on clinical trials for COPD patients. COPD hasn't stopped. We used to say it was the third biggest killer in the US. Sadly, COVID-19 overtook that a few weeks ago. So it's now the fourth biggest killer in the US. But we still need to do our research for COPD patients to keep treatments coming. Just for those of you who haven't met me before, very, very briefly, who are we? I'm part of a team that works down here at Harbor UCLA. We're professors, physiologists, nurses, PhD students, study coordinators, graduate students, and research assistants. And we've been here for over 20 years doing research in exercise and lung disease to try and help improve the quality of life for patients with chronic breathing difficulties. And as you probably know, we are kind of an exercise center of the universe almost. This terrible, horrible test that many of you may have done, the cardiopulmonary exercise test, where you have to cycle on a bike or walk on a treadmill and breathe through an apparatus, was invented by my academic grandparents at this campus. And we've done a lot of research on rehab, and this is a pretty good example of our rehab center right now, completely empty of people. We need to get back to being able to do that. We've published an awful lot, and I already told you we're responsible for developing the exercise test that's used clinically, but we also do a lot of studies with drugs like steroids, bronchodilators, oxygen, other medications. We've done studies with devices like non-invasive ventilations and oxygen delivery systems. And we're very, very interested, and we absolutely support and do a lot of research on exercise training and physical activity. Where are we? We're on the campus of Harbor UCLA Medical Center down Carson Street, where it meets the 110. And our building is over here on the campus. And this is our research dedicated building that currently has tons of parking. No problem with that right now. So I just wanted to share with you the impact in our center of COVID. This is, you may not be able to read this and it doesn't really matter, but I just wanted to show you how many projects we were running, most of them in COPD, but some of them in asthma, some of them in pulmonary hypertension, one of them in obesity. This is the number of studies we were running on March the 8th, and that's what happened on March the 9th this year, where only 2 studies remain open. These 2 studies are studies where we do phone calls with patients so we could keep those open. You know, COVID was devastating for us and for its impact on our ability to advance research and therapies for patients with pulmonary disease. And we've been scrambling ever since to try and bring these back online. The red ones are the ones that are closed, by the way. The green ones are open, if I didn't say that already. We want to bring these back online in a safe way. And 2, we want to contribute to the battle against COVID-19. And I'll talk about that in our later in the presentation. So how do you do pulmonary research during a pulmonary disease epidemic? Well, of course, safety in all research is the #1 priority. We can't, if we can't guarantee the safety of our subjects, we can't do research. We shut down our operations here on March the 9th, and I'm quite proud to say that in the context that we saw what was coming, we made the choice not to put patients at risk, and we shut down much earlier than even California, which was the first state to shut down, about 2 weeks later. And then we've been developing procedures to allow appropriate reopening of our clinical research, which includes waiting for what's happening in the local community transmission rate of COVID-19. We're not going to reopen until the transmission rate is low enough to make sure, to ensure that both the study participants and the staff in our center are safe. Of course, if you've been to the doctor or dentist recently, you'll be familiar with the procedures that they're using. They'll probably call you the day before and ask you questions about whether you've had any potential exposure or have any symptoms. We're doing that. They might ask you to wait in the car before your appointment and then call you to come in, which is what we're currently doing. Only one study participant is allowed in the building at a time. We screen people's temperature before entering the building, and we use face coverings and social distancing at all times. In addition, pulmonary research has a big difficulty, which is that we do procedures that are considered aerosol generating. And you know, you've listened to the news, right? You know that COVID is spread through aerosols, these tiny droplets that float in the air and can be breathed in by somebody else. So when you do a breathing test, like a spirometry or an exercise test, that generates aerosols and they stay in the air. And they could put people at risk if those aerosols carry COVID. So how do we get around this? Well, the first thing is that all staff doing aerosol generating procedure have to wear full PPE. And this is the sort of thing that we're talking about. Here's Robert and April in our lab getting dressed up to do an exercise test. Very different from what it used to look like. So we've got N95 masks, face shields, gowns, gloves, booties, hats, the works. We are requiring that all our study participants have a COVID test before they come to us, and that test be negative. At the moment, the requirement is within 5 days, and that's because it takes a long time to get the test results back, which is is unfortunate. We're hoping we can get a test sooner, and we're trying to source rapid testing that we can actually do in the building and get a result there and then. We use inline HEPA filters on all our respiratory equipment. If you don't know what that looks like, the little filter that you breathe through when you do your breathing test, that filters out just about everything and makes sure that nothing nasty gets into the breathing equipment, the measurement equipment. We have to disinfect our rooms after each assessment, which is a laborious process and slowing our research down. And we have to leave the room empty because of this issue about aerosols. The virus can be floating in the air for several hours after somebody with the virus has been in a room. So we have to leave our laboratories empty for those viruses, potential, if they are there, to fall to the floor so we can clean them, disinfect them. And we run a HEPA filter pump to try and clean the air quicker. So this really means we can only do about 1 test per day, when before we could do 10 tests per day. So it's really had a massive impact on us. We're working on installing a negative pressure, high air exchange clean room, which looks something like this. It would be a curtain system that goes across half the laboratory. And what that would do is mean that we could clean the air behind that curtain quicker, in half the time. All the airflow to the room would go through that clean area, and that we could clean the air out. So that would increase our turnaround time for doing research. So where do we stand right now? So clinical trials, and this was true nationally and as well as at our center, remained open for drugs that had potential direct patient benefit, especially for those that have no other therapeutic options. So for example, cancer therapies where the drugs are still under testing, but they might extend life, and there's no other treatments available. Those types of studies were allowed to continue during COVID because really the patients have no other options. If you are already enrolled in a clinical trial and taking a medication or using a device that would be potentially dangerous for you to stop, those types of trials were allowed to continue. So at our institution, they do a lot of research on HIV, and obviously stopping potentially beneficial drugs to prevent HIV would be dangerous for the subject. So those studies were allowed to continue. We remained open throughout for studies of COVID and SARS, CoV-2, that's the virus that causes the COVID disease. It took a while to get those studies up and running, and I'll tell you about a couple of them in a few minutes. In July, we reopened our center for study participants who have low risk for severe disease, like young subjects with no underlying health conditions. Right now, we're doing about 1 to 2 studies per week in young, otherwise healthy individuals. We've had no issues with COVID so far, but we're getting our center back up and running with this low risk population. And we'll reopen to all study participants when the COVID-19 community transfer reaches safe limits. Establishing what safe limits are is a little bit tricky, and I will talk about that later as well. But we're not going to reopen for patients with pulmonary disease until we're confident that we can do it safely. So what does the community infection look like right now? So this was a graph that's taken, I don't know if you've been using the LA County dashboard, but it's a very nice system. I got this data from August 16th a few days ago, and it shows you the number of cases per day in LA County. And around about here, early May, was when the lockdown was came off. You know, Governor Newsom said that we can start opening up a few stores and a few vendors and things like that, and people started to come out of their houses a little bit. And you could see that, you know, they thought it looks like it's stabilizing. Number of cases looks pretty flat. So we'll start tentatively to reopen. Well, we know what happened, right? Wasn't good. We reopened too early, in my opinion, and we suffered the consequences of that in Los Angeles County and also in California in general. On the bright side, the most recent data from this month is showing that cases are coming down, and this isn't because we're testing less. We've got high rates of testing. We haven't yet seen much of a drop in the number of people getting admitted in the local hospitals at Harbor UCLA, at Little Company. We're still getting quite high caseloads. But we're anticipating this drop in identified cases will translate to a drop in hospitalizations pretty soon locally. LA County as a whole, hospitalizations have already started to drop. This graph shows you the number of tests being done per day. That's the blue, light blue up here, the columns. And this solid navy line here shows you the average, what they call the test positivity rate. So out of all the tests done, what percentage of those tests result in a positive COVID test? So you can see that we're testing at relatively high levels right now. Back in May, when the release came off, we were testing at lower levels. The test positivity rate was about 5%. And the governor thought, okay, we're okay to let people back into the community. And that appears to have not been a wise thing to do. Now we're testing more, almost twice as much, actually, yeah, almost twice as much. And the test positivity rate is creeping back down towards 5%. When it gets to around about 4%, most of the CDC and the WHO guidelines say that's the level that's probably safe to start people getting back to some kind of normality. Well, doesn't mean that we shouldn't be using masks, doesn't mean we shouldn't be distancing, doesn't mean we shouldn't be washing our hands, but it might mean that we can go out more safely because you're less likely to encounter somebody who's positive. So that's what we're waiting for to reopen our center for higher risk patients for the test positivity rate to come down. So when we do reopen, what's coming? We've got a couple of very exciting studies that are coming online soon. I want to tell you about two of them. The first study is a very new concept, and we're very happy that we're one of the centers, the 21 centers in the US that will be doing this trial, and that we can offer it to the local community. It sounds a little bit dramatic, but actually you'll see that it's the early data are very promising. This was the original study that was done in moderate to severe COPD patients. So this is people with an FEV1 of around 70 to 30% range, so that middle range. And what happens in this procedure? So this is a bronchoscopic procedure where the patient has a bronchoscopy, and through the bronchoscope, a radio frequency electrode is passed. And what that electrode does is actually kill the nerves that are behind the airway. This is what it looks like afterwards, immediately afterwards. And then 3 months later, you can see there's no damage left at all. And I'm going to play you a little video, let's hope this works, that tells you about this procedure. Let's see here. Can everyone see and hear?
Yes, we can see and hear.
You or someone you know suffer from COPD or severe asthma? A new therapy called targeted lung denervation has been developed to treat these obstructive lung diseases. For most people with normal lung function, breathing is easy. The airways are wide open with minimal inflammation and mucus. When you suffer from a chronic respiratory disease, you may experience shortness of breath, wheezing, chest tightness, and a cough that produces mucus. Sometimes you may experience sudden flare-ups of these symptoms called exacerbations, which lead you to seek medical attention. Targeted lung denervation, or TLD, is a non-surgical procedure that interrupts overactive nerve signals and may reduce obstructive airway symptoms, helping you to breathe easier. A standard bronchoscope is passed through the patient's mouth and into the lungs, and a special catheter is passed through the bronchoscope to provide the treatment. Once the catheter is in place, the balloon is inflated. Notice the silver electrode on the inflated balloon. When activated, the electrode delivers a type of electrical energy called radio frequency energy, which penetrates to interrupt the nerves located just outside of the airways. Once the energy has been delivered in one position, the balloon is deflated and rotated to the next position. This is continued for 4 total activations. Upon completion, the catheter is removed, and the animation shows that the nerves distal to the treatment site have been interrupted, decreasing nerve signals throughout the lung on that side. The catheter is then placed in the lung on the opposite side, and the treatment is continued in the same 4 positions. The entire treatment is now complete.
Oops. Is that going to restart? I don't know. Well, there's only 30 seconds left. I'll stop there and go back to the slides. So the point of this is it's a little bit like a permanent, you guys all take bronchodilator medication, most of you, I imagine, you know, an inhaler, albuterol or Spiriva or something like that. So those drugs act on the nerves in the airways to dilate the airways. What this is doing is permanently interrupting those nerves so that the airways are always open without the need to take necessarily take extra medication. The early trial of this was done on 22 patients with quite severe COPD, and in 22 patients, the reduction in, this is the quality of life questionnaire called St. George's Respiratory Questionnaire. the reduction in that score, meaning that the patients felt better, was really dramatic and lasted out to a year. In a follow-up study, they studied 82 patients with this treatment. This study was published in one of the most important journals in respiratory medicine just at the end of last year. It recruited patients with severe COPD who are on triple therapy. So if you're on triple therapy, meaning bronchodilators and inhaled corticosteroids, the likelihood is that you have more flare-ups or COPD exacerbations than you're a frequent exacerbator, because those types of patients are put on this triple therapy. And this nerve denervation in the 82 patients more than halved the amount of people who had an exacerbation in the 6 months following up from the treatment. Both severe COPD exacerbations were reduced by the treatment and moderate to severe COPD exacerbations, although this wasn't quite significant. It's pretty amazing at the moment. We're quite excited about it. This study is going to be run at our center by Dr. Shah, who's the director of interventional bronchoscopy. You've probably seen him before. I think he's talked to your group several times. He's been a member of our team for a long time. We're not recruiting right now, but if you or anyone who is interested, we're more than happy to hear from you, and we can give you information when the study starts, when it's safe to start a study like this, you know, considering all the COVID issues we have to deal with. You can go to the Airflow trial website and get more information and see the rest of that video if you're interested. Obviously, it's a little bit more invasive than we're used to, this type of study. And it's targeted for the types of people that have the inflammation and lots of mucus production. It's not going to be very beneficial for people who have lots of emphysema. It's more for the inflammatory airways type individual. So anyway, so you're very welcome to go on their website or call us, and we can give you more information. Just so you know, we're not in the office, most of us, on a daily basis right now. So if you get an answer machine, leave a message. The messages are being listened to, and you will get a response. But the likelihood is you won't get someone to pick up when you call this number right now, because we're social distancing at work as well. Most people are working from home. The other trial that's coming up we expect to start in the next month or so, is quite pertinent for somebody mentioned Apple Watch earlier, that has the Apple are now putting a pulse oximeter in the Apple Watch. So we've been funded by the Tobacco Related Disease Research Foundation to develop a wearable sensor that might predict when COPD patients are going to have an exacerbation, in order to warn them to go and get treatment early.
Wow.
And this is in the development phase, and it's in collaboration with a great guy up at Caltech, and I'm very excited about this study. So you guys all know the COPD flare-up or exacerbation is typically due to getting an infection, and exacerbations are associated with accelerated decline in lung function, in physical function. You know that if you get hospitalized, you know, that puts you, you know, really hits you hard, and it's hard to recover and get back to where you were before. These data show that patients without exacerbations, their lung function declines over 4 years. But if you have one or more exacerbations, you decline more rapidly. So the key is to try and predict who's going to have exacerbations, prevent them from getting them, and or treat them early, right? So an exacerbation is the major cause of health decline, and it's the major cause of where the healthcare costs are in COPD. The big cost is, if you get hospitalized, and we're trying to avoid those. But symptoms of exacerbations are difficult to detect because they're very similar to what you feel every day. They are shortness of breath, increased mucus production, and sometimes you don't notice that you're getting an exacerbation is coming on until all of a sudden you're in the ER and then somebody says you've got to be admitted, and that's what we want to try and avoid. So how might we detect exacerbations early and perhaps signify patients to seek medical attention early and avoid severe complications? So our concept is to develop a sweat sensor, a sensor that you would wear probably around the wrist that would measure chemicals in your sweat that predicted signs of an oncoming COPD exacerbation. So this is my collaborator at Caltech. He's a biomedical engineer and a minor genius. And I was very fortunate that he invited me up to Caltech just before COVID hit, and I gave a presentation up there. This is me outside the world famous Beckman Institute. Well, what a beautiful place that is. It's amazing. Dr. Gao developed this platform, the concept of these sweat sensors. They're made from acetate and graphene, and they can be printed like printing words on a page. They're printed using a 3D printer, and they cost pennies, if not less than a penny to make. Each sensor contains a sensor for temperature because chemical reactions happen faster at higher temperatures, so we have to measure what the temperature is. It contains a module that actually induces sweat because most of us aren't sweating all the time. So it actually releases a little bit of a current into the skin and makes the skin under the sensor sweat so we can measure the sweat. It has a monitor for how rapidly the sweat comes out, and it sends a Bluetooth signal to a tablet or an iPhone app, reading out what the chemical sensors say. Right now, the batteries last about 2 or 3 days. And because these things are very cheap to produce, then we can just throw them away and use another one. So using sweat for diagnosing disease is not a new idea. In fact, it's currently the way that cystic fibrosis is diagnosed in children. Cystic fibrosis is caused by a mutation that alters the sodium and chloride concentration in the sweat, makes the sweat salty. And so this is how cystic fibrosis is actually diagnosed in little babies, that they put a little sweat inducer on them, they take the sweat out and they measure the sodium and the chloride. And you can see that Dr. Gao's sweat sensor can detect high levels of sodium and chloride in cystic fibrosis patients' sweat compared to healthy subjects. So proof of concept. But he's also developed methods to measure stress hormones. So cortisol is a stress hormone. It's released into the blood when we're under stress. And if you look at the concentration, this is nanograms of cortisol per milliliter of blood. It also gets through into the sweat. It's diluted by about 100 or 200 times, but it is measurable in the sweat. It's just very difficult to measure these tiny, tiny, tiny concentrations. But his sensors are sensitive. We recently developed one for an inflammatory molecule called CRP. Many of you may have had this measured by your physician. It's used quite commonly, and it's one of the inflammatory signals that the body releases when very, very early in an infection. Releases CRP, C-reactive protein is what that stands for. The body releases CRP into the bloodstream to tell the immune system, hey, we've got an infection coming. We need to get immune system cells to go and gobble up all those nasty bacteria that are invading the lung. So we can measure CRP in the sweat. This shows the concentration of CRP, very, very low concentrations in the sweat against the current produced on his sweat sensor. So we can, it's a linear, the sensor is linear and quantitative. We can determine the exact the amount of CRP in the sweat. And in a very early study, we used this just to show we could see difference even between smokers and non-smokers. If you're a non-smoker, the CRP in your sweat is slightly lower than if you're a smoker. These two blue ones are non-smokers, these two red ones are smokers. So even just somebody who smokes, not even got disease yet, we can tell the difference between them based on this inflammatory molecule. So the idea is that Dr. Gao will produce these sweat sensors. We will first go into the hospital to patients who are actually having exacerbations. This is what we would call a true positive test to see whether our sensor can measure inflammation in patients who are actually having an exacerbation in the sweat. So we would recruit people from the hospital. The study would take about 2 hours just at the bedside. We'd induce sweating, we'd collect a blood sample, and we'd compare the two. So that we would compare the blood concentration of inflammatory cytokines and with the sweat concentration. We're also going to collect saliva because that's another way we might be able to measure these biomarkers of inflammation. And hopefully the blood and the sweat will be linearly related, and that means we can use the sweat to calculate what the blood value would be. And then we'll measure them again after about 2 hours to see whether the sweat test that we did the first time and the sweat test we did the second time gives you the same answer. So that's how you determine accuracy and precision of a diagnostic test. These inflammatory markers, these indicators of stress, are not only released during infection, during pulmonary infection, prior to getting an exacerbation, they're also released during stressful events in your everyday life. So for a COPD patient, that might be doing some physical activity is stressful. So what we've built into the concept is an algorithm to try and determine when inflammatory markers are being released because the patient is doing some physical activity versus when inflammatory markers are released because they've got an infection. And we'll use the way that different biomarkers respond to either exercise or infection so that the watch can tell, oh, you don't need to rush to the hospital, it's just you're doing some exercise, versus, oh, there's a pulmonary infection coming, you should contact your doctor and get advice. So this is due to start in a couple of months, well, hopefully a month or two. We'll definitely be up and running by early 2021. And this study is funded by your tax dollars, the Tobacco-Related Disease Research Program, which is a California-based foundation. Just in the last few minutes, I'll talk about a couple of the COVID trials that are going on at the Lundquist Institute. So our institute, members of our institute are part of something called the ACTIVE Partnership. This is an acronym for Accelerating COVID-19 Therapeutic Interventions and Vaccines. You've probably heard on the news something called Operation Warp Speed. Well, the ACTIVE Partnership is the people are actually doing that. They're academic researchers, they're drug companies, they're FDA, they're the NIH, who are all working as fast as possible to try and develop therapies, vaccines, and diagnostic tests for COVID to help us get back to normal. The Operation Warp Speed aims to deliver 300 million doses of safe, effective vaccines by January 2021. We've got a long way to go, and January seems very soon, but I wish them the best of luck. In terms of diagnostic tests, I just told you about our biosensor for COPD that is funded. Well, of course, as soon as COVID came along, we pivoted our biosensors and wrote for a grant to fund it for sensing COVID. So this is the sensor that we've produced, a very mass producible printed biosensor, and we've tried it out. This is an inflammatory marker. This is called IgG. These are COVID positive patients. These are COVID negative patients. And this is a sample of saliva that we put on the sensor. We're actually measuring the protein, the antigen on the virus. So this tells you whether you've got virus or not in your sample. These are antibodies that the body produces against the virus. So IgM is very early produced antibody. IgG comes a few weeks later. And CRP is this inflammatory marker, tells you about how severe the disease is. So we think we've got a biosensor that can detect whether or not you're infectious, whether you've had COVID or not, and if you've got it, how severely you've got it. We can measure in 1 minute from a spit sample these variables. If you wait 10 minutes, the signals get even bigger. This is a 10-minute signal. And the really cool thing is that these sensors have a Bluetooth on them. They just go straight to an app on your mobile phone, and you don't need a special reader or anything like that. You don't have to do these PCR tests. They're sent direct to your phone. So you can imagine people who need to go to work. You just use one of these sensors every day. You spit on it in the morning. It tells you you're clear, and you can go to work. So we're hoping this will get funded. We will find out in about a month, and we can get on with doing this study. The Lundquist has been involved in several clinical trials of therapies for COVID-19 disease. I've just listed 2 here. I know there are others. I don't know the names of them all. There's lots of people working on this problem. It's hard to keep up. I mentioned the first one, hydroxychloroquine and azithromycin study, which is now closed. And of course, this is the drug that was very widely publicized. And I'm not going to make any comment on which side of the political aisle you fall, but it was very interesting to me that this study closed early. This study was a study of hydroxychloroquine, not in hospitalized patients, but in patients that didn't have such severe COVID, who were isolating at home. We called people to ring them up to see whether they would like to take this medication and potentially reduce their symptoms, and hopefully test whether the drug was useful for preventing people to get hospitalized with severe disease. And we got 2 answers. The 2 answers were, no, I don't want to be involved in that study. That's the drug that President Trump promoted, and I'm not interested in participating. Or, no, I don't want to be involved in that study. You're just trying to prove President Trump is wrong. I don't want to participate. So now we have no study. We don't know whether this drug actually might work for helping to keep people out of hospital. There is data showing the people hospitalized with COVID-19 that this drug does not work, but we don't know whether we give it to patients with less severe disease, it might be beneficial. So I just encourage people, please take part if you can, if you're confident, and if you're willing. This is how we get medical research done. We have a trial upcoming for a drug called Dapsone. This is an anti-inflammatory drug that's used for skin conditions normally. It has a good safety profile, and it's going to be used in patients who are tested positive for COVID-19 but are isolating at home, and we will call them and offer them to take this drug and hopefully prevent them from progressing to severe disease. Probably the most important trial that's coming up is the trial of the Oxford vaccine. This is the AstraZeneca vaccine. We'll start, our center is one of the places you can come and get vaccinated with this experimental vaccine that's coming in September. There's going to be 3 sites in LA that are participating in this, UCLA Westwood, us, and blanking on the third site. Maybe it's MLK, I've forgotten. But, you know, obviously vaccine is where we're all desperately hoping that we're going to make some progress. And the interesting thing about this vaccine, the vaccine trials, is actually they are trying to recruit patients who are most likely to get severe disease. So patients who are older, patients who have underlying conditions, because we will be able to tell more quickly in those people whether the vaccine is working. So in fact, patients with COPD, with other underlying health conditions will be selectively recruited into this trial. And there will be information coming. I'm sure you'll hear it and see adverts everywhere very soon about how to volunteer for this. We're not open yet, but we will be very shortly. And just to impress on you how incredible the global response has been. I don't know if you've been seeing any news reports about how long it typically takes to make a vaccine. So in January, if we started making, started identifying the need for a vaccine in January 2020, the normal route is about 2 years of academic research in Petri dishes with cells, and then about 2 and a bit years of preclinical research in animals. You then go into phase 1 trials in healthy individuals, phase 2 trials, phase 3 trials. And once you've shown that the drug works in a large population with multiple backgrounds of people, then you start building your factories to make the vaccine, which takes a long, long time, manufacture them, get them approved by the FDA and distribute them. 16 years is the typical timeline. This is what's happening for COVID. And the thing that's really impressive is that none of the steps in the normal academic through to phase 3 trials and approval by the FDA, none of the steps are being skipped. They are happening in parallel in order to speed them up. And another very impressive thing is that the pharmaceutical companies are taking an enormous gamble. They are building factories and manufacturing drug before it's even been proven to be safe or efficacious, in order that the minute that we find out there is one vaccine that works, that we can get it distributed to people right away. So I think you know that there are 6 vaccines that the US is trying to develop. Every site in the US that is in a vaccine trial is only allowed to participate in 1 vaccine trial. We don't want bias between a doctor saying, oh, you try this one and you try this one. So every site is only involved in 1 vaccine trial at a time. And we're involved in the AstraZeneca Oxford vaccine trial. And we'll tell you very soon when that is ready to recruit. So if you are interested to get involved, the best way to get involved is join our pulmonary patient registry. That just means calling our number that I gave you before, and we can distribute that. Right now, all we will do is give you a call and take your get you to answer a few short questions. When it's safe to come in and get a breathing test, then we will ask you to come in and get a breathing test. If you're part of our registry, it means that we have your name and number, and that we are allowed, you're giving us permission to call you when there's a clinical trial that you might be interested in or qualify for. That's all it does. So please call that number that I'll put up at the end, and if you're interested to participate in any of these things, so we can reach out to you when there's something that you qualify for. And if you don't like the sound of that, then please follow us at the Pulmonary Education Research Foundation. We run a free, I think you probably many of you know this, we run a free blog. We publish 1 or 2 blog articles per month and try and keep you updated with safety and health tips. Our address is perfsecondwind.org. We're all located down here in the South Bay, and we're a non-profit organization. And we're aiming to help patients with breathing difficulties. So please go to our website and just have a look and sign up for our blog. It's free. If you don't like it, you can easily withdraw and you don't have to receive it anymore. So thank you very much. That's all I wanted to tell you. And I'm happy to take questions. Let me see if...
First of all, a big round of applause for Dr. Rossiter, you guys, for an amazing presentation. Harry, before we get started, can you explain to the group a little bit about the Lundquist Institute?
Sure, yeah. So the Lundquist Institute, we used to be called LA Biomed a couple of years ago. Some of you might remember 20 years ago, we were called the Research and Education Institute. We're a private non-profit research institute on the campus of Harbor UCLA. We're all doctors at, all our members are either MDs or PhDs. The MDs are doctors at Harbor Hospital, Harbor General. We can't do research in the Harbor General building because it's a county building, right? We can't use county taxpayers' money to do private research, which is why the Lundquist Institute exists as a non-profit research institute for those UCLA professors to do their research down here in the South Bay. We've been here for, goodness, I don't know how long, 50 years, something like that. We're going strong. Does that help you, Kurt?
Yes, exactly. That's exactly what I wanted to know and hoped the group was interested in. So let's open it up to Q&A, you guys. Please raise your hand or type out a question in chat file. Edna, please go ahead. Edna, unmute yourself. Edna, unmute yourself and then ask the question.
Hi there. Oh, you flipped your camera too. Oh.
You're supposed to talk, Edna.
Where is Edna? I'm gonna try and unmute her.
The agreement's going. How are study agreements going? Somebody asked. I'm not sure I quite understand the question. Study agreements.
Dr. Rossiter, that was me. That's Carol. I just
Carol.
wanted to know where we stand.
Yes. So, oh God, this is like a long story. We are trying to get a written agreement between Little Company and the Lundquist Institute to allow me to come over to Little Company and recruit patients. Right now, Carol and Jackie can't give me your name and phone number because they'd be breaking HIPAA. If we have a written agreement between Little Company and our institute, then we can do that without breaking the law and directly recruit patients from PEP, from the rehab program. So we started about a year ago trying to get a contract, and it's just so complicated. You have no idea, lawyers getting in the way, but I am told that there is progress. But you've reminded me, Carol, that I need to chase that progress up because it's been about a month since I last heard from them. Thank you.
Let me know what I can do.
I will. Thanks so much. Thanks for your help so far too.
All right, can I ask my question now? Can anyone hear me? Oh,
yes,
okay.
Yes. Go ahead.
Yeah. Okay. Is the Lundquist Foundation founded or paid for by Melanie and Dick Lundquist?
That's correct. So about a year ago, we were called LA Biomed. Melanie and Richard Lundquist gave a bequest of $70 million on their 70th birthday.
Right.
Oh, wow.
$10 million
Yeah.
a year for 7 years is what they
Yeah.
promised.
They do a lot of that, yes.
So they get to name us. That's what that's for.
Well, they get to name the tower
Well, that's
over
okay.
in Torrance also.
Yes, yes, exactly. The Torrance Memorial Tower is named
Right.
on it. Yeah.
Harry, I have to unwind my brain and stop calling you guys LA Biomed now.
I keep doing it too, Kurt, so don't worry. I'm getting better. I've got the ribbon on here to
Remind you.
remind myself.
Some other questions from the group.
This is Jackie. I have a question. There's some really interesting things going on. On that bronchodilator one, where you go in with the bronchoscopy,
Yeah.
and deaden the tissue. Is this something? And I know the FEV ones are pretty severe, but are you going to be doing this on asthmatics as well? Because they seem to be the ones who have most of the bronchial spasms and on all those meds.
So I think the company is also looking at asthmatics. I don't think we're not involved in that trial, Jackie. But this trial will enroll patients between FEV1 between, I think it's 70 and 30%. So that's sort of mid-range. And there's lots and lots of inclusion and exclusion criteria, as there always are with these trials. But the reduction in exacerbations was unexpected and really dramatic. You know, you won't find a medicine, a pill that you can take out there that does what this looks like it's doing in that study of 80 people so far. So
fascinating. Also, that sweatband thing is just incredible. I mean, you have such great research and possibilities ahead for us. So thank you.
Yeah, I'm excited about the sweatband. So we're early days right now, but yes, if I can sell the idea to Apple, then I can retire. That's what.
Yeah, I'm your assistant. Good job.
Go ahead, Joseph, go ahead, please. Unmute yourself. You're not unmuted, Joseph. Push down on the space bar.
Nope, not yet.
If you're on your laptop, it's on the top. If you're on your iPad, it's on the top. You touch the top of the screen.
You should be able to unmute him as well.
Okay, let me try to unmute Joseph. Come on, Joseph. Let me go to
I'm not the only one that can't do this.
Okay, Joseph, hang on.
Oh, your son, he's good at it. He is. He's calling his son.
I have a question. The saliva, does that come through your mouth or your nose?
Mouth. So the
Okay.
everything that's in the blood, well, not everything, small chemicals that are in the blood can get into all your body fluids, right? So they could get into your saliva, in your mouth, they could get into something in the back of your nose, secretions, they could get into your sweat. So ideally, we'd like to, for the COPD exacerbation monitor, we'd like to have something you could wear around your wrist. I think that would be the most unobtrusive. But a backup plan is that we have something that you spit on every morning to test whether you're, how it's going. The COVID monitor, we want to measure the blood, the saliva, and also the nasal swab, because the virus lives in different places in the body, and we want to see whether we can detect it in those 3 body fluids and which one we can use. If we can use the saliva and still detect COVID, that would be the simplest thing to do. But a finger prick blood sample, like for a diabetic, would also be, you know, not too bad. Diabetics measure their finger prick blood every day, right? So there's lots of hands up. Who wants to go next?
Can you hear me?
I can hear Rita. Is that Rita?
Yes, that's me. The last time I was in the hospital, I was fine in the afternoon. I was talking to my neighbors. My daughters came over with dinner, so we're talking maybe 2 or 3 hours. And the next thing I know, it's 12 days later, they're taking me off a ventilator. I didn't even know I was sick. I mean, how would this help me? And how would I know that that's happening?
Well, so I think that's the point. It's very hard to spot a pulmonary infection before the body reacts to it very severely, like it did for you, right? So you probably had bacteria in your lungs that were starting to cause an infection for a day or two before you were rushed to hospital.
Even though I had no cough, no fever, no...
Even though you had no signs or symptoms, right? I mean, did you measure your temperature every morning?
Not every morning.
No.
But even when I was admitted to the hospital, I didn't have a fever.
Right.
They just
yeah, and so these, but your blood, your inflammatory system, your defense system in the body would have been responding during those couple of days before.
Wow.
To try and defend against that infection that you had.
Well, they said my O2 level was 122. Does that make sense to anybody?
No.
CO2?
CO2, that's what, yeah.
CO2. Oh, okay.
Okay.
Right. Yeah, so that would make, yes, that would make you hard to remember things, I imagine.
But how would I know
Go ahead, Rita.
How would I know that's happening until I end up in the ventilator?
Well, right now it's very hard to know that it's happening, Rita. That's the whole point. So we're trying to come up with ways to detect it in the day or two beforehand, so you could go to your primary care physician and get antibiotics and not end up on the ventilator, right? So if you combated that infection earlier, maybe you wouldn't have gone that far. That's the idea. Whether this idea works, I don't know. This is an idea at this point, but we're gonna...
Sign me up.
We've managed to unmute Joseph. Joseph is...
I think I got on there. Can you hear me?
And then Roberta.
Can you hear me now?
Yes.
Yes. We can hear you.
You can?
Yes.
Yes.
So my question was, you said they were working on 6 different vaccines for COVID. Is that correct?
I think in the US, the government team identified 6 different vaccines early that they would support. Yes.
So
$2 billion each.
Okay. So just a question: Are they going to narrow that down to where we have a choice of 3 or something, or just one coming out of that, or?
If all 6 work, I'm sure that they will use all 6. So there are different mechanisms of different types of vaccine. In other words, one's an RNA vaccine, one's a denuded virus, you know. So there's different types of vaccines. If they all work, I'm sure they'll sell them all. Some will be safer than others, some will be more efficacious than others. So some may not work at all.
Right. So who decides which one, like we would get in California here?
I don't know that far away. All I know is that so right now there's a vaccine trial, and I've forgotten the name of the vaccine. It begins with an M, I think, that you can, if you want to go up to West LAVA, you can sign up for it. It's current, it's active, and it's running. It's recruiting subjects. And there's another clinic somewhere up near downtown LA that's doing that trial. We were selected to do the AZ trial. We're doing that one. And once the AZ trial is finished, we're trying to recruit 500 people to the AstraZeneca vaccine trial. They have to be COVID negative, obviously, because we need to see whether they're going to catch it or not.
Yes.
So they get COVID tested, and then they'll get it's a 2-dose vaccine. You have to come in twice to get it. And we're trying to recruit 500 people in 2 months. That's how quickly we're trying to do this study. Once we finish that one, maybe we'll be selected to be a site for a different vaccine, but they're not doing them at the same time in case the doctors are accidentally biased the study. Yeah. but
We
once
we
they're found to be safe and efficacious, I don't know who decides where the vaccines go and who gets them. I'm positive they will go for frontline workers first. The doctors that are putting themselves at risk every day by going into COVID wards will be right up front on getting the vaccine, I'm sure. And then
I have a question.
high-risk patients will be next.
We have time for a few more questions. And Yvonne has been in the queue the longest, so Yvonne is next, and then Archie.
Wait, we've got Roberta and Bibi also have questions.
Okay. Go ahead, Yvonne. Everyone, just one question, please, because we're running a little bit over time for Dr. Rossiter, and he's got to get back to doing his good research.
Dr. Rossiter, if the wristwatch measuring sweat study is successful, could this potentially be something to be used for those of us who don't have COPD but may have another form of ILD, IPF, or just pulmonary fibrosis?
I think the idea that it could be used as an early warning sign for pulmonary infections in general, I think that's a reasonable idea. So, you know, if you're prone to getting pulmonary infections, whatever disease you have wouldn't necessarily have to be COPD. I agree.
Okay, Archie, go ahead.
Roberta, we'll go ahead and do Roberta next.
Okay, Roberta, then Archie.
I was just reading yesterday about an Israeli, I don't know what to call it, a discovery. They have some kind of a mouthwash to determine whether or not you have COVID. Apparently, you rinse your mouth twice with this mouthwash, and it has worked. It has had a proper answer 80 trials out of 80. So
wow.
I'm wondering who's making sure that the American doing the sweat study is comparable to the Israeli doing a saliva study.
I don't think there's, well, so there's a website called clinicaltrials.gov. I don't know if you've heard of that or been there. It tells you all the clinical trials that are going on in the country. And if you're doing a clinical trial, you have to put your study on there. So one of the things that we do to make sure that we're not doubling up work that other people are doing is go to that website and look for what else is happening in the world. There is already, I've seen there's already one approved for saliva. It's a PCR test, so it means you spit in something and then the saliva gets sent away and gives you a result. So we're not the only people doing this. Our sensor is a little bit different because it's called a multiplex. It actually measures 4 different things in one sensor. Whereas these other sensors are only measuring one thing. They're measuring the virus. So frankly, at this point, you know, we need more options. So if somebody else develops a mouthwash, if somebody else develops something else, I'm all for it. I have no problem with that.
Well, the question is, who's the clerk of the works? Who is making sure that these people know about each other's work, or just each person's responsible for seeing if there's a clinical trial?
Somewhat, yeah. So somewhat, but the active consortium that I told you about in my talk is trying to police that a little bit. So the FDA, the NIH, they all got together and they said, you know, every drug trial has got to come through us, so we know what's going on. We don't want 50 sites in the country all doing the same study. We want 50 different studies on 50 different things. So there is some level of organization going on, but different countries, it's hard to, you know, it's hard to follow. But thank you hadn't heard about the Israeli one. I'm going to look it up as soon as I get off.
Okay.
Okay,
Go ahead.
Doctor. This is Archie, KD6OLH, commonly called the old Lucky Ham. I saw in your slides you talked about exercise and its effect on COPD persons. Did you mention anything about exercise and other things that were going on? Like, I'm not COPD, I don't have too much worry about the lung thing, but I found out that my exercise did a lot of help for my cancer and the other things. Can you give me a more enlightenment on that? What other things that the exercise can help with, please?
So we call exercise as medicine, right? And exercise has so many benefits on so many things in the body. It has benefits obviously on the muscles, the heart, the lungs, because that's what you're using when you do exercise. But you know, when you do regular exercise, you actually release chemicals into the body that travel around the bloodstream and have benefits in other places. So we know that people who do regular exercise actually have better protective cognitive function. So their brain is working better. We're actually doing a study right now in young, healthy men where we're trying to determine whether exercise benefits the genes in the sperm. So what you pass on to your children is actually benefited by being fit versus fat. That's what the study will determine. It helps the liver, it helps the kidneys, it has an anti-inflammatory effect. It actually reduces damaging inflammation in the body. And in fact, it releases anti-cancer chemicals into the blood. So there was a beautiful study done about a year ago. It was an animal study. They put tumors into a mouse, and some of the mice, they gave them running wheels and allowed them to exercise train, and some of the mice they didn't. And the mice that were exercise trained, the tumors were smaller than the mice that were not exercise trained. Because exercise releases anti-inflammatory things into the bloodstream, and then cancer is an inflammatory disease. So it actually retarded the growth of the tumor.
Thank you, doctor.
You're welcome.
He has a question.
BB?
Yes, it's the on the 500 subjects that you're going to have for your test. Are there going to be a percentage of them that get a placebo, or will you know you have the actual drug?
The vaccine trial, I don't know the answer to that. Once the advertising goes out, there will be full information about what the trial exactly is. But the advertising has not yet been approved, so I can't tell you anything about the trial because I don't know it.
Okay. Thank you.
We have time for one more question, and then we've got to let Dr. Rossiter go.
Roberta had her hand up.
Oh, you already got me.
Oh, we did? Okay, sorry.
Roberta. Who
Drink
else?
more wine.
Who else? Any other questions?
No.
Well, let's give Dr. Rossiter a huge round of applause. Huh?
Thanks so much. I love seeing you all. This is just wonderful. I want to see you in person next time.
It's so great to see you, Harry. Thanks so much.
And thank you for taking time out of your schedule to be with us today, Dr. Rossiter.
You're very welcome.
Very
interesting, very interesting.
Let's thank Jackie for lining up Dr. Rossiter. Please give Jackie.
Yes, Jackie.
Thank
you.
Jackie.
Thank you. And next month we're going to have, I think next month we're going to have my therapist, because you know I'll go to therapy, I'm talking about coping with the pandemic. How do we feel about being closed up? So that will be our next guest speaker. And I think after that, I think Dr. Stringer wanted to speak to you guys.
Okay.
So we really are going to have some really good people come in and talk to you guys. Okay?
All right.
Okay.
Great lineup of speakers.
Yeah.
Thank
Love
you.
you guys. I love you
Thank
all.
you.
It's so good to see you guys. It's big
Yeah,
day. Bye, Dr. Rossiter. Love and kisses to everybody.
Bye, everybody.
Bye.
Bye. Take care.
Take
Okay,
care.
bye.
Adios. Okay.
Hi, Carol.
Recording. Stop the recording.